In this video, we will discuss about in vivo results about the synergistic effect of 5-FU and HTPBD TRAP1. Before directly going into the in vivo results, the researchers has presented some of the in vitro results that are done on the organoid which are developed from this APC mice. So what they did is they take the 18-year-old mice that has this colorectal cancers and then took this harvest this intestinal adenoma and then seed them in matrigel and grow this colorectal cancer in the organoid and then treat this organoid with different conditions. That is one with 5-FU, one with HTPBD TRAP1, and the third condition is HTPBD TRAP1 plus 5-FU. And in the panel B, we can see that number of organoids are decreased in the conditions where HTPBD, TRAP1 and 5-FU is used compared to the all three conditions. And these things they have validated using the immunofluorescence image, where we can see that the pink fluorescence also goes down at minimum when they use HTPBD plus 5-FU, indicating that amount of organoids is going down in this case, and also the proliferations markers, that is Ki67, which is used, is also goes down. The quantifications of this organoid count is shown in the panel D, where we can see that organoid counts is high where neither 5-FU or neither HTPBD trap is used, whereas when both the reagents are used, both the drugs and 5-FU and the nanoparticles with HTPBD nanoparticles with TRAP1 plasmids is used, we can see a minimum number of organoid growth. And the Ki67's results are also quantified, where we can see that less proliferation markers when both of them are used, 5-FU and HTPBD TRAP1. Once validating their in vitro results, they decided to check the in vivo effects, synergistic effect of 5-FU and HTPBD TRAP1. So they use this orthopedic injections of the tumors, and they inject the 5 × 10⁵ tumors per mouse, and then after 7 days they inject this HTPBD and other conditions, that is HTPBD plus 5-FU and HTPBD trap plus 5-FU, and after 5th days, so at 0 days they oral inject this HTPBD, and after 5 days they intravenously inject 5-FU and CSA. CSA is the inhibitors for this mitochondrial membrane mitochondrial permeability transitions, and at 10th day they analyze the cells, and what they are able to see that when they use this HTPBD plus 5-FU, they are able to see a decrease in the fluorescence intensity, indicating the decrease in the tumors, whereas when they use CSA plus HTPBD trap plus 5-FU, they see an increase in the intensity, indicating that CSA inhibit the mitochondrial inhibit the damage to this mitochondrial permeability transitions. And when they in order to check the and these things, the panel G results are quantified in the panel H, where we can see that the minimum bioluminescence is shown where HTPBD and 5-FU is used. And they also validate their specificity using a Western blot, and they can see that in the normal tissues they do not see a decrease what we can say that in the case of this HTPBD and 5-FU in the tumor tissues, they see a increase in the knockdown. The panel I is about this Western blots results of the normal tissues and the tumor tissues, and they can see that in the case of normal tissues, they do not see an increase in this Western blot signals, whereas in the case of tumor tissues they see an increase in these Western blot signals, and these Western blot signals corresponds to This Western blot signals corresponds to this uh, tumor growth. This Western blot signals corresponds to this tumor growth, which is kind of minimum in the case of normal tissues, but a maximum in the case of I need to check whether this panel I Western blots corresponds to which uh, protein of interest. Let's move on to the J part where they check this proliferation markers, and the TRAP1, we can see that TRAP1 goes down when they use HTPBD and 5FU, but it goes up when they use CSA along with them. And similarly, the proliferation marker goes down when they use HTPBD TRAP1 plus 5FU, but it goes up when they use CSA along with it. And thereafter, they check the tumor bioluminescence, which is minimum for the case where they use HTPBD plus 5FU. And when they check the survival rate, we can see that survival rates are slightly higher, or it goes up to 45 days for the case where HTPBD and 5FU is used, indicating that it will, when both of them are together used, it will suppress the growth rate of the suppress or delays the growth rate of the tumors. So I hope you understood the results that are presented in this figure. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. So that's all for this video. And regarding this panel I will mention in the descriptions what this Western blots belongs to. Uh, uh, one of the guess that I can make is that it shows this HTPBD and 5FU. I'm not able to recall that Western blots. I will update the results in the descriptions. But thank you, and that's all for this video.